Alex Hopper. I'm an undergraduate research fellow here at Northern Michigan University, and I'm going to be the interim moderator for the next hour and for the next 4 presenters. And because we're a little behind schedule, we're just going to get started with our first student researcher is Cora Suda, who is a biology and zoology concentration major. Her mentor is Dr. Kurt Galbraith, and her title is New Pika Tapeworm Samples from Mongolia Add Support for Their History of 2 Intercontinental Colonizations and Reveal Previously Unknown Clades.
Good afternoon. Today I'll be talking about how pika tapeworms can inform us about intercontinental exchange.
That's not where you just hit the space bar.
So we start off with Beringia. The Bering Land Bridge connected North America to Asia and allowed different species to cross across that connection and create animal diversity in North America. And our goal is to understand the history of this connectedness. So our species of interest are pikas. They're small lagomorphs, which are relatives to rabbits, that are found on the rocky talus slopes and high elevations of western North America and of Asia. In many pikas, there are tapeworms, more specifically the Schizorchis tapeworms. These tapeworms can give an insight into the biogeographical history of pikas. It's hypothesized that pikas came from North America came from Asia to North America in 2 waves of colonization, and their tapeworms can give an insight on the timing of this colonization. A previous study published in 2020 proposed that 2 colonizations occurred approximately 10 million years ago and 4 million years ago. This study was based on DNA that was sequenced at 150 different locations in the nuclear genome called loci. The more loci you have sequenced, the more the nuclear genome you have to create relationships among species. What this study lacked was species diversity from Asia. The inclusion of this diversity has the potential to change the colonization history. In order to come to a more complete analysis, data from more recently collected samples needs to be added to see if it adds to this diversity. This is the host distribution from the original study. As you can see that there's quite a few host samples in North America, and then there's only 2 in Asia. So we acquired 16 specimens from varying locations in Mongolia, and this added 4 more pika hosts. The 4 new ones are the ones that are in bold. And even though we have 8 host samplings, there is a total of 34 different kinds of pika. So this only covers a small portion of the host diversity. Each of the different triangles represents a different Schizorchis lineage. And as you can see, one of our questions was, why does Ochotona princeps have 4 different lineages? So that's definitely something that we have to look into. From the 16 specimens we collected, we got DNA sequence data for all 16. I designed 22 nuclear loci primers, and I had 11 show strong amplification for the DNA. With those 11 primers, I ran polymerase chain reactions to multiply the DNA, and then I visualized the products with gel electrophoresis. The samples that showed strong amplification were sent for DNA sequencing, which is done through an electropherogram, which shows you different colors for the different nucleotide bases. I then aligned the returned DNA sequences and conducted an in-depth Bayesian analysis through the software StarBeast, which allows for the independent evolutionary histories of each gene region to be used to reconstruct the single overall population of the Schizorchis genus. This software finds the most common shared history between the different species. So this analysis software goes through and it creates all these different kinds of phylogenetic trees for each loci region, and then it compiles all of them into the most common species tree. The resulting tree created 4 genetically diverse clades that were previously unknown in the data set. The new clades are denoted by the colored branches, and the different colors indicate a different host. The colonization timing fell around the same time as the published work, 8 million years ago and 4 million years ago. The place of colonization is denoted by the orange arrows. The blue bars indicate 95% probability of the node age. This means that the new lineages added did not change the interpretation of the previous work, and the 2 colonizations is still consistent. Each of the new clades have greater than 99% probability of occurrence, besides the one clade on here that does not show a circle right here, species 2, but that had a previous question in the previous study as well. And although the samples aren't comprised of 150 loci, they still add to the data set and they form a meaningful contribution. These new samples continue to support the narrative of the 2 colonization events by picas and their Schizorchis tapeworms. However, there's clearly diversity in Schizorchis that hasn't yet been described. The new samples created 4 new Schizorchis clades and new hosts to the overall analysis. But once again, that's only 8 hosts of the 34 possible. Therefore, to continue, there's more sampling to be done to keep adding to the data diversity from other pica species, and the new Schizorchis samples will need morphological identification to confirm their species. And that's it. Does anybody have any questions?
Can you go back to your gel electrophoresis slide? So, when you're doing a gel, you said you had to send in samples to where?
So when we get the PCR product through the gel electrophoresis, we then send it to a different lab. I believe it's in California, because they have the electropharogram there, and they run.
Is it the gel itself that you're sending on? Or are you looking for things on the gel?
So you're looking for the amplification on the gels, and so if it shows a strong amplification, then your PCR product is good to send to that lab.
So you're talking about sampling more picas. Are there any regions you think would be particularly valuable to get more samples from?
I feel like there was a lot of samples taken from North America, so I definitely feel like more Asian diversity would be a wise addition.
So when you're looking at additional tapeworms from Asia, are those ones that were isolated at the time of the original sample collection, or do you go through preserved gut samples of tapeworms?
I guess I'm confused by your question.
Where do you get the tapeworms you were analyzing?
So Professor Galbraith took a trip to Mongolia after his publication in 2020, and then he also took a trip in 2022, the summer months, and so the samples that I added were from the first trip and then the trip in 2022.
Worms are taken out of the guts in the field.
Okay. Does anybody have any other questions? Okay.
What's your favorite type of pica?
I mean, there's quite a variety. I don't know if I could pick a favorite. Okay, I think then we're good.
I drag this over. Okay. I don't need it.
Is the dongle plugged in for this?
Sorry, get out of your way.
I suppose that would help.
Okay, you the next presenter? I sure am. Okay. I'll just introduce you in like a minute. I'll introduce you in like a minute that you can plug. He has his own computer. Can everything be plugged into it? Okay. Yeah. Okay. Okay, the next student researcher is named Xander Fontella, and he is an environmental studies and sustainability major and has a history minor. His mentor is Dr. Sarah Mittelfelt, and his title is Filling Empty Lands, Hands and Stomachs, or a close look at the triumph of the 1893 Pingree Potato Patch Plan and its implications for the present. And just plug those in. I don't want to close it.
Okay. All right. We ready? Okay. Good afternoon, everybody. I am Xander B. Fontella. I'm here to talk to you about the original community gardening program in American history and why I think it's still very important today. Before I begin, I want to give this dedication to my beloved Aunt Jackie, who could not be here to witness me do this, but I know she'd be very proud. So, anyone who knows me, you know how much I love living in Detroit. It's many things to many people, but to me it's home, and I'm very proud of that. And it's not at all like what you people make it out to be up here. It's a very lovely city. But that being said, it's not what it used to be by any stretch. It is hard to see on the empty lands and prairies about what kind of places these used to hum here. Factories, neighborhoods, you name it. And despite its recent advances, Detroit has a lot to go. It still loses majority of its tax basis to urban sprawl, can barely efficiently fund its transit and other city amenities, and if you don't have a car, it's really hard to live there. As someone who lives there without a car, it's hard to live there. And this is legacies of redlining, automobile culture, and white flight that has been ongoing since the late 60s and into the 20th century. So in the 2010s, Detroit did embrace urban agriculture, and it created international headlines about addressing food insecurity in urban settings. But we've been here before, and to act like that we haven't I find very disingenuous. And going back all the way to the 1800s, 1890s even, Detroit managed to create the very first program of its kind. And it's all thanks to a man named Pingree. So if you've been in Detroit, you drove down Woodward Avenue past Grand Circus Park, you may have seen the statue of this bald, white, fat gentleman. And if you don't know who that is, I don't blame you, because many people don't. On his plaque, it reads in many things. Hazen S. Pingree calls him a gallant soldier, enterprising, successful citizen, 4 times elected mayor of Detroit, and twice elected governor of Michigan. And in big capital letters, idol of the people. Now, that is a very bold title for someone who, again, many of you have never heard before until just now. But I can tell you, and I can go on and on. In fact, I had to cut 5 minutes out of this presentation going on about how much of an amazing person this guy was and how much of an outlier he was for other politicians of his kind in his day. But going back to the statue, I happened, when I was there and visiting, I managed to leave a few potatoes as a little bit of respect to the guy. So, about the man himself, Hazen Stewart Pingree was a Yankee shoemaker from New England, born 1840, 1901. And while he spent many of his time in Maine, he was a Civil War veteran who fought in the First Virginia campaign and decided to move to Detroit after hearing about its splendor from his other infantrymen. In moving to Detroit, he became the second most successful footwear company of his kind. He was also a beloved social and municipal reformer who spent much of his time as mayor fighting corporations in office and lowering prices for working people and being an advocate for many working people of Detroit, especially immigrants. In 1893, there was an economic panic that caused all sorts of trouble across the nation, but in Detroit, especially worsening employment rates, especially affecting the immigrant Polish variety, growing hunger and civil unrestlessness, and large swaths of empty land was going undeveloped from the lack of economic development. So as the story goes, Pingree was riding through the empty subdivision one day and was noticing all the empty land and what could possibly be used to grow with it, or what could possibly be done with it. And after a talk, allegedly, as the story goes, with his wife, who suggested originally to convert the land as gardens for the unemployed, he decided quickly to pitch this idea to the public. That didn't go well. The city laughed at him pretty much. People thought he was kidding at first about how this is impossible, this is ridiculous. The press lampooned him, even foreshadowing his run as governor and what design of the state seal might look like, such as tuber instead of tubor. I thought that was really funny. Then, because of this press, suburbanites feared potato bug invasions, and many people refused to help him, even the churches. He wrote to the churches appealing to their humanity, such as on the left here, you can see his appeal to them, who they only gave him $13.80 for the entire program. And he spent his entire next 3 terms fighting the churches and aiming for their taxation. So as a way of showing out against the city and pushing back against these calls of ridiculousness and how serious he was, he decided to put up his horse for auction as a means to pay for his potato patch plan. This was the point when many people saw how serious he was and decided to donate, and this is when many donations and funds began to pour in. Now he began to assemble and get to work with his agriculture committee. So of the 3,000 people that signed up in the first year, 945 families received plots, and these plots consisted of anywhere from a half acre to a quarter acre. Gardeners received all necessary tools, seeds, information to get started, and agricultural committee even consisted of Captain Cornelius Gardner, who oversaw growing of barrack gardens in Fort Wayne the year prior. Following data is from the report from 1896 at its height. And you can see here that from the initial, only $2,600 more or less was spent at its year's height. And from an initial budget of $4,000. And you can see that in the output, the sheer amount of money that was developed and grown from the harvest developed to be $30,998, which is nothing to scoff at. Over the first 3 years of its run, you can see this upward pattern of budgets being saved and a continuous uprising of funds, and many of these funds actually went towards social and municipal programs, saving taxpayers money. No? Oh, hello? Okay. Pingree publishes 5 conclusions in a magazine, being that, number 1, people will always help themselves if given the option. Effective use of idle land can provide lots of food for lots of people. Families can grow sufficient food from even the most smallest of plots. Landowners would much rather have their lands be utilized than have to pay heavy taxes on it. And the best way to help those in need is to help humanize them first. And many of these things are not lost upon today. Here's a map of Detroit picturing all the different plots across the city, showing the numbers numbers adjacent to them being the amount of people per plot. Here overlaid a modern city of Detroit. You can see just where many of these places were. And in modernity, many of these places were converted to park and green spaces after they were done. This was the first of its kind, too. Many people in the press wrote about how no other English-speaking country or city has ever decided to do a program like this. And it created traction interest across the country. Here's a map of generally the idea of major cities across the country that developed similar plans, such as the Pingree Potato Patch Plan. And as you can see here, going from Portland to Portland, also Berlin and Toronto. So this made international news, and many people were uplifted from his programs. So this begs the question, what went wrong? So Pingree decided, when Pingree couldn't be mayor and governor at the same time, he decided to be governor, and many of his ideas went with him. And then as he spent time fighting the corporations at a state level, he unexpectedly died in 1901 after succumbing to an infection he got on a safari with his son. Even though it was already on its way out by the time he left governorship, this was the symbolic end of its run. So the year is 2024, and Detroit is a very different city. It's post-industrial, very modern metropolis, and there's a lot going on between the largest demographics. Where it used to be a majority European, Yankee city, now it's majority Black, Latino, and Arab. And while public transit is much more expensive, population is higher, unemployment rate is higher, and it also faces different things in urban agriculture, such as the tendency of being co-opted for gentrification. And there's a lot happening already. So people would try to argue that maybe we don't need a potato patch plan. But let's say we did, and what would a similar plan look like today? Given the amount of educational nonprofits in the city of Detroit today that are working to carry legacies of the potato patch plan, whether they know it or not, it would be really interesting to try to get these organizations to work together with the city, get them talking and using history to inform the present, community self-empowerment, culturally relevant produce, and prioritizing the needs of people who live in the city already would bring a lot of good to the city and help a lot of people. Thank you. Any questions?
All right, so a large portion of what you focused on was the funding, the source of funding. Where do you see, if this would be implemented again, or a form of, where do you think the majority of the money would come from?
So the original funding came from city donors, it came from grants as well as private donations. Many of the original, like in the first year, was private grants, and then as the program gained success and notoriety, city began to become more generous in giving grants, allocating funds for this. But yeah, I think as far as today goes, we probably have there's a lot of funding already for urban agriculture in Detroit, and there's a lot of many sort of independent groups working together, doing their own thing, working towards the similar goal. So trying to unite these groups, maybe pool that money together that each going towards these little different programs, could making a super program using the information and history of the potato patch plan of 1893 could, I think, would do a lot of attracting ideas and interest for something like that.
You indicated that in the historic example, it kind of expanded and then fizzled. Where are we in the current cycle that began around 2010?
So as it kind of stands, it's becoming more of a, since 2010, it's becoming more of an institution. With many, as the city has become the city has wanted nothing to do with it pretty much. While they've been more laissez-faire about it, they have not indicated any actual plans to create any agricultural plan. They're leaving up to the people to do it for themselves. So at this point, we're kind of at the point of where enough guerrilla gardeners have kind of gotten their gardens that are getting up traction and showing that they're working, that these things are working, that we're kind of, how do we make this less of it's now trying to normalize it at this point, instead of trying to, because we already have the thing in place, people are seeing how successful it is, how do we keep it? How do you keep this fire from going out, basically? Because what happened in the original plan is that it just, as the administration lacked interest, as Pingree left the office, there was also, not to mention that there was also an economic boom happening right after in the 1900s. So there was definitely sort of loss of a need for this kind of program. Whereas now we're seeing that there is a need for it, and that a lot of the similar aspects of it can be addressed with something like this. So, anyone else?
So I know around 1913, Henry Ford actually gave profit sharing to his employees because the economy was doing so good. So was it when the economy started doing good they realized they did not need this?
Pretty much, yeah. Because this was to address economic depression, the panic of '93. So because many of the people lost their jobs, land wasn't getting developed, so you had to do something with these people in the land. So that's what this was.
Was it implemented after factories started shutting down in Detroit, or was the idea brought back up?
This is the first time this is being brought up since, because there's not a lot of academic literature on this. There's passing mentions, you look at any academics about gardening in Detroit, you might see Pingree's name, but you're not going to see any detailed operations about what the ins and outs of it and why it was so successful. You might see it worked and it was good, but that's the end of it pretty much. So I'm trying to give it this program its credit and to show that we don't need to reinvent the wheel for solutions as perennial as food insecurity. When we look at the past, and sometimes it's just as simple as giving people the means to grow their own food in the ground where they live. So, anything else?
How come at the time of the Great Depression, they didn't try to bring this back?
Well, actually, it's funny you bring it up, actually, because they did do something similar. There's something called thrift gardens that they tried with similar ways. It was more of a community effort than a city effort, but it was sort of like in the same model of like people without the ability of getting their own food or groceries, who were hungry, could go to these gardens and like put in work and get food out of it. It was sort of kind of like a co-op program, that unlike the Potato Patch Plan, where it was more of like a your plot is your plot. It's in the 30s, it became more of like a do as much work, take as much as you need to. So the legacies continue of the plan, but it never was actually realized in its entirety. Anything else? Thank you so much. And then here are my sources.
Oh, okay. Let me plug this in and do the introduction. Does it not automatically pop up anymore?
Just kidding.
Do you want to use your own laptop?
If you don't mind.
Okay.
Yeah, no, that's totally doable. Should I drag this over?
What we're doing.
Set that up. So as soon as that's done, you might just have to drag it over and just make sure that your display is set up for extended display, or at least duplicate, so it'll pop up onto this.
That's you, right?
Yeah, that's me.
Okay. So the next student presenter is named Dustin Goodman. He is a medicinal plant chemistry major, and his mentor is Dr. Leslie Putman, and his title of his research is Quantification of Cannabinoids and PFAS in Erlina hemp plants treated with PFOS and PFOA.
Hello, my name is Dustin Goodman, and I'll be going over the quantification of cannabinoids and PFAS in Erlina hemp plants treated with perfluorooctane sulfonate and perfluorooctanoic acid. So PFAS is per and polyfluoroalkyl substances. There are thousands of different PFAS molecules. Both PFOA and PFOS are perfluoral. This means that all of the hydrogens attached to the carbon backbone are replaced with fluorine. There are poly PFAS molecules, which means only some of the hydrogens are replaced with fluorine. Some common applications of PFAS is nonstick pans, waterproofing, and stain proofing. PFOA and PFOS are two of the most studied and have the most concern of PFAS molecules. Possible side effects of PFOA include kidney cancer, testicular cancer, and high cholesterol. Possible side effects of PFOS include reproductive toxicity, immune effects, and kidney toxicity. We don't have enough evidence to say that all PFAS molecules are toxic, as there are thousands of them. There is not a lot of research done on all of them, but we are exposed to PFAS molecules due to ingestion and biosolids applied to farm fields and landfill leachate. And PFAS molecules are sometimes referred to as forever chemicals, or they are referred to as forever chemicals, and this is due to the carbon-fluorine bond being very strong, so they don't break down easily. And the objective of my study was to treat plants with 2 different concentrations of PFOA and 2 different concentrations of PFOS to see how much the plants actually took up. So to start this study, I sterilized Erlina FC8 hemp seeds. I germinated them on a paper towel for 9 days. They were then planted in pots with ProMix, and they were grown in an indoor growing facility and split into 4 treatment groups and a control group. 2 of the treatment groups were PFOA, 2.5 and 5 micrograms per liter, and the other 2 were PFOS treated with 250 and 500 micrograms per liter. These plants were watered every day with 200 mils of the PFOS solution made with aquaponic water, and the controls were watered with unaltered aquaponic water. After 91 days, the plants were harvested and separated. I separated the aerials, the stems, the roots, and seeds, and then they were stored in a minus 80-degree freezer. The stems and the roots were extracted using methanol, and they were both ground using a mechanical grinder. The aerials and seeds were pulverized using a mortar and pestle and as well extracted with methanol. And the soil was ground using a coffee grinder, so it would get turned to a fine powder and be easily extracted as well using methanol. The aerial tissues and seed extract were analyzed using gas chromatography paired with mass spectrometry to quantify cannabinoids, delta-9 tetrahydrocannabinol and cannabidiol, and to the plant tissues, seed, soil, and, all of the plant tissues, the seed and the soil were all extracted, and they were, the PFOS and PFOA was quantified using liquid chromatography paired with tandem mass spectrometry. So here we have our CBD in the seeds for all of the plants. There was no THC seen in the seeds at all. You can see right here that the 2.5 and the 5 microgram per liter PFOA seem to have the highest concentration of CBD, and the others were relatively similar. And over here, we have our milligram CBD per gram of plant tissue for the CBD as well as THC here. We have the blue as the control, the orange and the green is the 2.5 and the 5 microgram per liter PFOA, and the blue and the purple is the 250 and 500 microgram per liter PFOS. And for the THC and CBD, there wasn't much of a difference between all 5 groups. So, and then here we have the uptake of PFOA and PFOS in the aerial tissue. And as you can see, even though the plants were treated with 100 times the amount of PFOS, the plants took up 200 times the amount of PFOA. So the 2.5 microgram per liter PFOA was slightly higher. The plant took up slightly more of that than it did the 5. And over here we have the plant taking up less of the 250 microgram per liter PFOS as it took up more of the 500. In the roots, we as well still see that there is a very high uptake of PFOA. There's 10 times the amount, and the plants took up 10 times the amount of PFOA than they did the PFOS. And as you see, the lower concentrations treatment groups pulled up less than the higher concentrations. And then here we have the stem as well, following the same pattern. The PFOA took up, or the plants took up 100 times the amount of PFOA than they did the PFOS. This time, again, the 2.5 microgram per liter PFOA treatment group pulled up more than the 5 did, but the 250, it has stayed the same the entire time. The lower concentration treatment took up less than the higher concentration. There was no PFOS. Well, there was a very small amount of PFOS. It was below the limit of quantification, so the only thing that I could show was the PFOA. The seeds, there was a decent amount of PFOA taken up into the seeds, as you can see here. And so after I harvested the plants, I ended up going back to the soil and taking 3 bores of the soil and grinding it up to see how much PFOA and PFOS was still in the soil. And about half PFOS, there was roughly half as there was the PFOA. So there was still more PFOA in the soil than there was PFOS, even though the plants, as I mentioned, have were treated with a much higher concentration of PFOS than they were PFOA. So for my conclusions, cannabinoids are relatively unaffected by PFOS and PFOA contaminants. The plants took up exponentially more PFOA than it did PFOS. 200 times more PFOA was taken up by the aerial tissue, 100 more into the stem tissue, and 10 times in the roots, even though the plants received 100 times the amount of PFOS. The plant took up more PFOS as the concentration increased, but the opposite was true for the PFOA, except what we saw for the stem tissue. Leftover PFOS in the soil was approximately half of the PFOA, and the concentration, for PFOA, the concentration was highest in the aerial tissue, and then the stem, then the roots, and the soil. For the PFOS, the highest concentration was in the soil, and then the roots, the aerials, and then followed by the stems. For future studies, I would like to analyze hemp plants periodically through the growth cycle to see if there is a limit to the amount of PFOS molecules that these plants can take up. I would like to analyze the plant extracts for degradation products of PFOS to see if the hemp plants are actually breaking down the PFOS. I would like to acknowledge my fellow researchers, Asbel Wells and Luke Horn, and Dr. Putman, as she has supplied me with all of the PFOS that I used in my research project. And that is it. Dr. Nguyen?
Have you tried to germinate the seeds that you know have PFOA or PFOS in them?
I have not. Because so all the seeds were ground up, so I didn't save any of the seeds, but I would be curious to see what happens if that was done. Dr. Bodman, Dr. Canfield?
Do you have any idea the pH of the soil, and if yes or no, do you think that would have had an impact on the sulfonate and the acid?
I would assume there would be an effect. I didn't test the pH for the soil throughout the study at all, but that would be interesting to test it in a future study. Excuse me. Dr. Bodman?
I'm a non-science major here, so don't hate me. Can the cannabis plant be consumed by humans after this has happened, or not?
With it taking up so much of the PFOS, I would say no.
Okay. And then why was the PFAS taken up more than the PFOA?
That is something me and Dr. Putman are still discussing.
Okay, so that was a good question.
Because yeah, I would like to know that as well, because yes, it got 100 times more of the PFOS, but for every group there was vastly more PFOA.
Thank you.
Joey?
Do you know if there's like a remediation process that could get rid of this, or is it just like?
So part of the project was kind of looking into the phytoremediation. I've talked to Dr. Putman about if these plants were incinerated, it would get rid of the PFOS molecules, but it's just not economically sound to do. Dr. Chinelli? Oh.
So do you think that there's like an upper limit? So you saw with the PFOS there was, more put in the soil, the more the plants were taking. Do you think there's like an upper limit where you can like saturate the plant?
Me and Dr. Putman have discussed that. I think so. I have no idea what that saturation point would be, but that's why I would like to look into that into the future to see, because doing the 250 and 500, I assumed that it would be in the sky, but there was actually more of the PFOA, so I don't know. I would assume that the saturation point, so to speak, of the PFOA would obviously be higher than the PFOS, so I would definitely like to see that as well.
Yeah, two quick questions. How many plants were in each group is question one. And then for the within group variation, was that genotype, or what do you predict caused the variation within a group?
So they were set in the tent differently. So to answer your first question, 3. There were 5 groups, 15 plants total, 3 plants in each group. I mean, there could be like maybe the grow setup. I mean, the plants in the first row may have been getting different amounts of light than the second and the third row. The wind, they were all the same Erlina FC8, so I don't think that would have an effect, but I'm not sure entirely. Dr. Nguyen?
Two quick questions. So first, do you have a source actually that's saying that
Dr. Putman.
You do?
That's my source.
You know? I'm just curious if that's been confirmed in the literature yet.
Yeah.
It has? Okay.
I was pretty sure, but I wasn't 100% positive.
Temperature and things like that.
Got it. Okay, and then the second was, is how do the concentrations that you were giving to your plants compare to maybe contamination sources, like the leachate from the waste management kind of stuff?
So I know that 250 to 500 is, I'm assuming, much larger. I know me and Dr. Putman in our typical studies just do 2.5 and 5. But I want to say the safe, like what they call the safe is, I think, in the parts per billion, correct? Parts per trillion, excuse me. So it's definitely low as to what it should be. Dr. Wilson?
Yeah.
I feel like am I going overtime?
Yeah, that's probably the last question.
Okay, sorry.
Make it a good one.
Go, please.
No. Yeah, I can ask you later.
Yeah, I mean, I'll be here. We can like walk outside and talk as
Please go
well.
ahead and ask a question.
He had one too.
Sorry, I just didn't want to take up all the time.
All right. 2 questions?
So
Shut down.
do you have a hypothesis what's happening to the PFOS? Is it just not extractable in the material, or is it being destroyed, or is there an experimental design flaw here?
I think
Where'd it go?
the PFAS is getting degraded.
Okay.
But that's why I want to look into that in the future, because that's what me and Dr. Putman have hypothesized on her studies. And doing this, I mean, I feel like this proves it even more that it's obviously going somewhere. So yeah, I really would like to look into that to see if that's why. Like, you know, obviously there's much more PFOA in the plants than there was PFOS.
What year are you?
I'm a senior.
You say you graduate?
Yeah, I graduate this May.
What, good. How much time roughly on average per week did you spend doing all this? The total experiment was 5 months?
So last semester, most of the time was just spent watering. So I mean, it was very minimal. You know, I made the solutions once a week, and then maybe like 20 minutes
Including
every
all
3
the
days.
Spectroscopy and.
Oh no, so that was done this semester. I spent,
Do you have a total calculation of hours spent?
no, it's a lot.
I'd be curious to learn
Yeah.
that. If you had an unlimited budget and you were completely unhinged, what would you do? To understand this. The bottomless budget, unhinged. You have no regulation, no nothing. What would you do?
I mean, keep growing plants and trying different experimental designs to see. I mean, first off, I want to see what's happening to the PFOS, but I also would like to know why the plant took up so much more of the PFOA. I mean, to get 100 times less and still take up 200, 100, 10 times more, it was very surprising when I saw that. So I would like to look into that more. I mean, really just keep running different experiments to see where it's going, how it's getting pulled up, even using maybe PFBA, a different PFAS molecule, and looking more into that as well. But just doing research, I guess, with just continuing it in any way I can.
Cool, thank you.
Thank you, guys.
I think I emailed it to you, so you should.
Think that what?
I think I emailed my presentation to you guys, so you should have it.
Is this your presentation right here? Okay.
Oh, I actually have.
Yeah, I don't know. Sorry. I accidentally got to your email somehow, so.
Doesn't help that I have 10 million tabs open, so. Alright. That one, and then right up. I'm just gonna do that. There we go. Beautiful. Yep, let's just exit out of that. We're going to minimize that. Get out of that. So that's the other one that you said we're gonna get rid of that. That's the only other one that you need.
The next student presenter is Mary Kelly. She is an environmental science major with a French minor?
Double major.
Double major, okay. Her faculty mentor is Dr. Matthew Van Grinsven, and her title of our project is Use of Aerial LiDAR Survey to Support Restoration and Management Objectives for Mitigation Wetlands in Marquette, Michigan, United States of America.
I know it's F11, but I don't know how to. Sorry, should have asked you before. Behind here. All right, where's my mouse? There it is. There we go. Thank you. Sorry. You're good.
Thank you so much. Well, excuse me. Well, good afternoon, everybody. My name is Mary Kelly. I'm a senior here at Northern, environmental science major, and this is my project focusing on the use of an aerial LiDAR, or light detection and ranging survey, to support restoration and management objectives for mitigation wetlands here in Marquette. So this project, this work I'm presenting this afternoon is part of a larger project that began back in 2011, and that started as a partnership between the City of Marquette, the Marquette County Conservation District, and NMU, and together these three entities constructed 4 mitigation or man-made wetlands to fulfill a state of Michigan permit to restore around 4 acres of forested or natural wetlands. And so since 2011, the Conservation District, as well as NMU students and faculty, have continuously monitored this area for native and non-native species. The annual monitoring usually involves that of common tansy, reed canary grass, Canadian thistle, and many, many more. And of those, common tansy and reed canary grass are the most aggressive invaders in the wetlands. So there were 2 objectives of the project. The one that I'm talking about today is sort of the short-term objective, and that is to create a high-resolution digital elevation model, or DEM, and topographic wetness index, or TWI, using a LiDAR survey. Quickly, TWI is an estimate of where water might accumulate in an area given differences in elevation and slope. It's often used to assess flood risk in land use planning, for example. So a city might delineate areas that are subject to flash flooding and figure out where or where not to build things in that area. And it's also used in determining vegetation patterns, which is kind of another long-term implication of this work. The second and more long-term goal is to integrate that DEM and wetness index with any existing hydrological and vegetation data from prior monitoring. There's about a decade of prior monitoring to compare these 4 wetlands and improve those restoration outcomes. So since I am graduating in 2 weeks, my hope is that whoever carries on this work is able to sort of accomplish that second goal and really bring the project full circle. So our study area, very close to campus, Presque Isle Mitigation Wetland Area. Some folks know this as the Presque Isle Bog, but it is not a bog. It is a mitigation wetland area. And so on the left here, outlined in white and labeled, are the 4 mitigation wetlands. And we also have Lake Superior to the north and the south. So, methods. In the fall of 2022, we went out to Presque Isle and did this LiDAR survey over kind of the wetland area and a bit more. So it's around 29 acres that encompasses these 4 acres of mitigation wetlands. And this flight consisted of a UAV, so an unmanned aerial vehicle, and or a drone, plus a LiDAR sensor and a red, green, blue sensor. And so this setup allowed us to not just get that LiDAR data that we're looking for, but also get some visible light images as well. we collected the data at an altitude of 50 meters above the ground and got 3 returns. So quickly, how LiDAR works for anyone in the room who's not familiar, the sensor I was talking about shoots a laser down at the ground to collect that data while in motion. The drone kind of goes back in a back and forth kind of square pattern, and that reflected time data combined with the GPS system on board provides X, Y, and Z coordinates of that laser target. So you get kind of a 3D representation of whatever is there in space. So the returns, so we got 3 returns, those are the results of that emitted laser pulse. So they're usually numbered in the order in which they hit objects below. So typically the first return is the tallest object in that area. So in the case of a wetland, it might be like a really big tree. If it's a parking lot, maybe it's a person. And the last return is typically, but not always, the ground, because it's the ground. And soon after that data collection, we reconstructed it into a point cloud using a special software, DJI Terra, and imported that into ArcGIS Pro, so a geographic information system, sort of computer mapping software to classify and extract those points that represent the ground elevation below. I then converted those points into a digital elevation model with a spatial resolution of 0.5 meter. And from that, I was able to derive some hydrology features like flow direction and flow accumulation slope. And then plugging those values into kind of the GIS calculator of sorts resulted in the wetness index. So our first result here is our digital elevation model. And our ground elevation, you can see, ranges from around 146 to 152 meters above sea level. The darker green areas of the map are those of higher elevation. And then the mitigation wetlands are outlined and labeled for your viewing pleasure. We also have the wetness index itself. So these values ranged from less than 1 all the way up to 25. The darker green spots on the map in like B, C, and A indicate higher TWI. So those are areas of where water has a higher potential to accumulate. So those are kind of your wet spots. The low TWI, so those lighter areas of the map, are spots that have a greater potential to contribute water. So those are going to be drier because the water is leaving. And so I noticed also that while making the map, we have wetland D, which is a much lighter color. It has a much lower TWI visually than the other 3. And this led me to my next goal, which was to see how these 4 areas sort of compared to each other. So I started off with some stats. I did a single factor ANOVA test to determine if statistically significant differences in mean TWI exist in general between the 4 wetlands. And this revealed that yes, they do exist. We can observe for wetland D that the average is much, much lower than the other 3. However, this test does not tell me which groups in particular are different from each other. It simply tells me that not all of the means here are equal. And so I continued on with a Tukey-Kramer test, and the goal here was to determine which wetlands experience those significant differences in wetness when compared to one another. And so here we observe kind of in those highlighted areas, comparing wetland D to A, B, and C, that we observe that significantly lower, wetness amongst those 4 wetlands. So at this point, given the DEM and the TWI developed during the project, we might hypothesize that in wetland D, we might have more establishment of common tansy. Common tansy does well in drier environments, and since wetland D is dry, we might have more common tansy. Conversely, with reed canary grass, less likely to establish in D because it enjoys those wetter environments. So we're looking for kind of a higher TWI in wetland B or C for that reed canary grass. And then kind of moving forward a bit, the Conservation District might use these different maps to sort of guide their future monitoring. So using these to determine specific areas of concern, and thereby helping to lower risk of future invasive species establishment. More work could focus on different portions of this 28-acre space, since we've only looked at 4 of those acres. I also mentioned the decade of vegetation data. That could be looked into as well, with other students who might carry on this work as I'm leaving. And again, so just the project has the potential to inform regional conservation efforts when combined with the work of future students. And I'm really looking forward to seeing how the Conservation District uses this in the future. Maybe not while I'm here, but with other folks carrying on the work. Acknowledging these folks for their support. Thank you. Okay, questions? Yeah.
Well, of all the different computational and/or field steps, what were maybe one of the more challenging aspects? What you had to
Ooh. Well, I guess working with the LiDAR data to begin with is something I had never done before. And those data sets are also huge. So it took a long time to make the maps in ARC because the data sets are so big. So kind of time, I guess in terms of time, processing time for data was hard. And also just, I don't do a lot of statistics. So the ANOVA and the Tukey-Kramer, I had to do a bit of research to figure out if those were the right things to do. And then figuring out how to do them was a bit challenging, but it ended up being fun in the end, so good stuff. Yeah. Yeah.
So that TWI, that's not necessarily how wet it is, that's just the potential for wetness based on the
Yes.
geographical slope.
Yeah, it's just a measure of kind of potential for wetness, yeah.
Must be a pretty sensitive system for depth of a meter.
Yes.
How high was it gonna go?
Oh gosh.
Ish?
I'm not exactly sure. But that,
Must have been a treaty.
yeah. Yeah. Yes, Randy.
What about seasonal variation? Is it
Ooh.
important for future students to go out at different seasons, or does that matter for your questions?
I would think so. I mean, we get different levels of precipitation at different times of the year. So, I mean, we did this in October. So like maybe doing something in the summer, when things are a bit more like leafy, or I suppose you might be able to do like a winter study. So I guess precipitation would play a big role in the seasonality of it, yeah. Anybody else? Yeah.
Do you think these research areas are going to be pretty homogeneous, or do you think there's like edge effects? Like it looked like there's a road or something.
Yeah.
Road.
Let me pull up that map again. So, oh gosh.
Yeah, like that dry curve.
Oh, right up here. Yeah, so that, in theory, if I had the time, I would have kind of cut out that part because that is just the road. So yeah, I suppose in some spots it could be rather homogeneous, but yeah, there's room for improvement on the maps for sure. Yeah. Okay. Thanks, everybody.
Well, thank you everyone for coming out and supporting these presenters. It means a lot to them. Hopefully next year we'll be able to build on this and have even more. It'll be exciting. Thank you for coming out.